In this video lecture, we're going to start talking about the steps of the progress of an infectious disease in the human body. And the first step, as we mentioned on the last video clip, is that a microorganism has to enter the body. I know that's kind of sounds like common sense. So any location or any route that microorganisms can use into the body is considered to be a portal of entry. So you've got a diagram over here that is showing you some various locations that can serve as portals of entry. Now just keep in mind also that not all pathogens use the same portal of entry, and they generally have to come in the correct portal of entry so that they can get to the part of the body where they actually cause disease. So microbes arrive to the body through direct or indirect contact, and we'll be talking about that more toward the end of the semester. Now this is going to be true for pathogens as well as our transient and resident microbiota that we talked about during the first video clip. And again, in order to have an infection, you're becoming harmed. So an infection is caused by some sort of a pathogen. Now it can be a true pathogen or an opportunistic pathogen, but those types of organisms, they have to overcome the first lines of defense in the body, which we'll learn more about when we get into chapter 12, but in general those are going to be your skin and your mucous membrane linings that we've talked about. Those are the barriers to entry of microorganisms into deeper parts of the body. They have to do that, and then they have to become established and start multiplying. So the skin and the mucous membranes will be common sites of entry anywhere you have a hole, an opening to the outside. We'll talk a little bit shortly about the parenteral, and it is parenteral, it's not parental, route of entry. And then also crossing the placenta, we'll talk about that as well when you have a developing embryo or fetus in the womb, and the placenta is the site of exchange of gases and nutrients and wastes between the mother's blood and the blood of the developing embryo or fetus. Some microorganisms are able to cross from the mother's blood to the blood of the developing embryo or fetus, unfortunately. All right, so what about the skin? So we said pathogens are going to have to overcome barriers in order to cause an infection. So any kind of breach in the skin will be a portal of entry for microorganisms. When you guys take Biology 201, ANP1, you learn that the skin consists of an epidermis layer up at the top. That's right here on this diagram. And as you go down deeper, you have the dermis. That's your thicker, deeper layer of the skin. And the epidermis, that's where your barrier really exists, because in there you have very tightly packed epithelial cells. There aren't very many, there's not much space in between the individual cells. And you have keratin. Remember the keratin on the outside, the protein fibers called keratin that create the kind of waxy, waterproof barrier at the very surface of the skin. So those features prevent the entry of most microorganisms. Even though it's very thin, most microorganisms can't get past that. Now anything that causes a break in the skin, like you see here, can give microorganisms access down to the dermis. And the dermis is generally thought to be free of microorganisms. We don't have normal microbiota down here in the dermis. So when microbes get down in here, you can develop an infection. Now some microbes are able to come through your sweat glands. They enter that way, or the hair follicles, the sebaceous, the oil-producing glands in the skin as well. And in fact, when you have acne, that's due to bacteria that have infected a sebaceous gland. All right, and what about the mucous membrane? So the mucous membrane linings of your respiratory, digestive, urinary, and reproductive tracts are also barriers to entry of microorganisms. And now they generally are, it's an epithelial lining like you guys learn about in ANP1, so you've got tightly packed cells there, so microorganisms generally can't get past that unless they have some sort of ability to do so, which we'll learn about later, but most microorganisms cannot, as long as that mucous membrane has not been pierced in some way, as long as it's not broken. As an example of a mucous membrane barrier, so this is supposed to be a chunk through the inside lining of the trachea, and you guys might have learned a little bit about that in your A&P1 and or A&P2 class as well. So if you study the inside lining of the trachea, you've got a bunch of cells in there that are jam-packed together and kind of wrapped around each other. That's called a pseudostratified epithelium, which means falsely layered. It looks like a multi-layered group of cells, but it's actually a single layer, and they're just all twisted around each other and look like there are multiple layers there. And you've got some goblet cells. You guys remember those from A&P1? Those are cells that produce mucus. And in the trachea along the inside lining, and also in your nasal cavity as well, the epithelial lining there has cilia along the surface. And what the cilia do, if you've got microorganisms that are coming down into your trachea, the mucus there will trap dust particles and microorganisms that are moving down in there, and then the cilia sweep them up and outward. So it's preventing microorganisms from moving down deeper into the respiratory tract and down into your bronchi and even deeper down into the lungs where you don't want microorganisms growing. So that's those are some features of the mucous membrane linings of parts of your respiratory tract. Mucous membrane linings are generally thinner than the skin, and they lack the keratin, so they don't have those keratin fibers that provide that nice, tight, waterproof barrier. You probably would not want a waterproof barrier on a mucous membrane inside the body. I don't think dry mucous membranes are something that are very pleasant. But the mucus is there to help trap microbes and other types of particles from invading deeper in the body. Some other mucous membrane barriers in the digestive tract, that stomach acid is a very good barrier, a protective feature. You know, so many microorganisms, every time you swallow food or water or just your saliva or whatever, you're swallowing microorganisms, and they go sliding down the esophagus and they splash into your stomach, and that stomach acid that's there, that hydrochloric acid, is going to kill almost everything that lands there. So that's and in fact even the cilia in your trachea here, they sweep microbes up and out, and then you're either going to cough them out or you may swallow them, so they wind up getting murdered by your stomach acid. So that's something people don't realize right off the bat about your stomach acid, is that it's actually playing a really important role in helping to control the numbers of microorganisms that are invading the body. Okay, how about the parenteral route of entry? So parenteral is referring to microorganisms entering deeper tissues of the body due to a break in the skin or a mucous membrane. So anything that breaks the skin or mucous membrane allows entry of microorganisms is considered the parenteral route. So it could be a bite, a stab wound, some kind of deep cuts. This happens during surgery. If you get an infection following surgery, that's a type of parenteral route of entry. Yes, I know this is a weird picture of somebody whose hand was bitten by somebody with really big canines. Look how big those canine marks are. They're kind of scary. Might have been a vampire. Crossing the placenta. When you guys take Biology 202, you learn about the placenta. And the placenta is an organ in the uterus that has the mother's blood passing through it. So it's got like little cavities where the mother's blood passes through. And then these blood vessels that you see here, here's the umbilical cord. So you've got fetal arteries and fetal veins passing back and forth between the placenta and the developing fetus. And so the fetus's blood comes up here and travels into the placenta, and then you have this very thin membrane, this very thin wall of tissue that separates the mother's blood from the blood of the fetus that's passing through the fetal blood vessels. So that very thin tissue there allows oxygen gas to come into the fetus's blood. It allows the carbon dioxide and other wastes to go out so the mother's blood can carry it away. Nutrients are transported, things like glucose are transported from the mother's blood into the fetal blood vessels, and then that's carried back to the fetus through the umbilical cord. But that little thin wall, the mother's blood and the blood of the embryo or the fetus are not supposed to mix with each other, because you could have incompatible blood types. So you don't want any mixing. You need a little thin wall of tissue there that allows exchange of gases and nutrients and wastes. So even though there's no physical contact, there are some microorganisms that can get across that little thin wall of tissue, and unfortunately there's some pretty nasty things that can do so. One is a Toxoplasma gondii, and that's a protozoan that causes toxoplasmosis, and cats are a host for this, and the cats shed cysts of Toxoplasma gondii in their feces. So you've probably heard that pregnant women are not supposed to change the cat litter, and that is because if they get exposed to Toxoplasma gondii and it gets inside their bodies, it can cross the placenta, go into the developing fetus, and can cause things like fetal brain damage. So not a good idea. Toxoplasma is something we probably get exposed to quite a bit, but if you have a normal, healthy immune system, you just fight it off. The immune system wipes it out, and you don't even know that Toxoplasma was ever inside your body. But if you are pregnant, your immune system is weaker, so you would be more likely to develop an infection with that. And even if the mother doesn't have any symptoms of toxoplasmosis, it can still cross the placenta and go into the fetus and cause severe problems. Treponema pallidum, that's a spirochete type of bacteria that causes syphilis. That can cross the placenta, unfortunately. HIV, unfortunately, can cross the placenta from a mother to a developing embryo or fetus. So unfortunately, there's some pretty nasty things that are capable of crossing the placenta. Not all microorganisms can. So just because a mother has an infection with some kind of bacterial organism or a protozoan or a virus, not all microbes can cross the placenta. All right, there's a table in your textbook. Just take a look at it. It's showing you for particular portals of entry, what organisms commonly use those portals of entry to gain access in the body, and they show you how access is gained. Like, for example, Staphylococcus aureus and Streptococcus pyogenes gain access through the skin, through nicks and abrasions and punctures, so these kind of parenteral routes of entry. Herpes simplex type 1 enters around the mucous membranes of the lips, and so forth and so on. Some things enter the body through insect bites. So just take a look at this, not because you need to memorize it, but you want to develop an appreciation for the fact that different pathogens use different portals of entry, and they gain access in different ways to the human body. Another concept with entry is what number of microbes is coming in through that portal of entry. That is, there is a minimum number of microbes that are required to cause an infection. That's called the infectious dose. So it's not like, for most microbes, most pathogens, just getting exposed to one viral particle or one cell of a microbe is not going to be enough to establish an infection. There's a certain dose that's generally required. And that there's a value that has been calculated or estimated for many pathogens that's called the ID50 value, and that stands for infectious dose 50%. So if you had a room full of people and they all got exposed to the ID50, whatever that dose may be, it could be 10 cells of some bacterium, it might be 100, it might be 1,000, whatever it is for a particular organism, that dose would cause disease in 50% of the people in the room. The other 50%, the immune system would wipe it out and they would never have any symptoms of the disease. So that's called the ID50 value. There's another value called the LD50, which stands for the lethal dose. So that's the dose that will kill 50% of the people or the hosts exposed to it. Now over here, this is a little figure which is showing you some different microorganisms categorized according to virulence. More virulent ones at the top, less virulent down at the bottom. The more virulent a microbe is, the lower the infectious dose. The less virulent an organism it is, the higher the infectious dose. So these organisms up here at the top, like Francisella tularensis, which causes a disease known as rabbit fever, I think the infectious dose, 50% for that is around 10. That's a pretty low number of individual bacterial cells that you would have to get exposed to before you wind up getting sick from that. Yersinia pestis, which causes the plague. You've probably heard of the bubonic plague and the pneumonic plague before. That's a pretty highly virulent organism. Now down here at the bottom, these are things that are really often part of our normal microbiota and things that we get exposed to all the time. And these are not virulent organisms, and you would have to get exposed to a tremendous dose of these in order for them to actually be able to harm you. So higher infectious dose down here, lower infectious dose up here. Check your textbook for other examples, because they do provide a couple of examples of infectious pathogens for which the infectious dose is believed to be as low as 1 bacterial cell or viral particle. All right, so that's entry into the body. The next video clip, we're going to talk about step 2. So microorganisms come into the body. That happens all the time. And a pathogen has to attach to cells or a tissue somewhere in the body and stay in place so that they don't get flushed out by normal bodily processes. And once they do that, that's going to provide an opportunity for them to multiply and increase their numbers. So step 2 will be attaching to the host. That's a critical aspect of the ability of a microbe to cause disease and make us sick.